Hofmann. I'm employed at the TU Dortmund University in Germany, and I'm the last speaker for the session, which means I'm standing in between you and your lunch, so I hope we can get quickly through the slides. I'm also here on behalf of a close colleague of mine, Dr. Ioan Muntean from the Technical University of Cluj-Napoca. He, due to a very unfortunate situation at his university, he was not able to come here over to the US in person, so he asked me whether I could give this talk on his behalf, and I agreed, so I'm here. And I try my best, unless a large briefing. I'm not familiar with every detail, but nevertheless, I'll try to present the research as accurate and correct as possible. So Ioan is a group leader at this institute, which is mainly responsible for grid support and middleware support. Of course, they are also dealing with Globus, and they recently did some support efforts for two different research communities. One is the medical research and the other the computational neuroscientists. And this is what this talk is about the results of those two different areas. So let's step into the first part of the talk, the in silico cancer, biomarker discovery and validation. The initial motivation for this research was to derive a more sophisticated and personalized treatment for patients. If you consider you have a medical problem like the prostate cancer, nowadays the treatment is pretty much relying on medical statistics. If you have a medicine, and there have to be large studies before those medical stuff can be used for the public use. And the idea here was to somehow integrate more information, indicating more details about the condition and the stage of the patient. They're so-called biomarkers that help to diagnose also and to derive a more sophisticated personalized treatment. All right, so those biomarkers are mostly not known at the moment, so there has to be a process to be set up, the biomarker discovery and validation process, which doing in a traditional way, which would imply typically large studies and clinical trials. But the idea is now that they could be greatly improved by analyzing and correlating findings from the so-called multi-omics area, such as proteomics, genomics, and so on. And biomarkers are, of course, different gene expressions, altered or mutated genes, microRNA, and also correlations between those. Herein, you face a lot of problems, a lot of challenges of this multi-omics data overlay, because you have to somehow analyze and find correlations. One is that you have to understand the biological implications of this. You have also to process large amounts of experiments because there are a lot of genes you have to take into account. And there is a high level of complexity regarding the integration. You have different data sources, you have different quality of the data, and so on. All right, so this is how the in silico biomarker discovery and verification process looks like. At first you have a medical problem, and now the task is to find out for this particular medical problem which biomarkers are relevant. Then if we take the prostate cancer example into account, you usually conduct a tissue analysis, so you extract the genes, proteins, microRNA in order to analyze them. And the first step here is to somehow detect abnormalities. There are some biological values, such as the p-value or the folder change value, that tells you whether this gene or protein is deemed as abnormal. So you end up with a basic list of meaningful genes and proteins, and those data is now in the next step being compared with other data from the areas of genomics and proteomics using a couple of standard databases. Standard databases that are curated by the community, by the medical community, and they also contain validated data, which means it's of high quality. After that, they do an additional statistical filtering, And the problem is now how to link those correlations and biomarkers together with specific diseases. Therefore, you need to have a sort of biological knowledge extraction algorithms. And to do this, they rely pretty much on the PubMed database, which is a very popular database containing a lot of publications from the area of medicine. And after that, those information is again cross-validated by a sort of this biological knowledge discovery databases. So this process is being mapped to the following system, which the colleagues in Cluj have been developed. It is a multi-layer architecture application framework and consists of a presentation layer, of course, where the user is able to input the relevant data. It contains of a service layer in order to query and to activate statistics filtering services and so on. It contains also a processing layer with the data mining algorithms, statistics, matching algorithms, and so on. We have the integration layer I've recently talked about with a lot of heterogeneous databases that have to be integrated and consolidated. And of course, we need a sort of remote layer which controls the access to the grid and to the cloud processing, which is pretty generic. All right, so let's have a more detailed look into the second part, which means this is this part here. So what they are basically doing in the PubMed is they have the specific candidates and retrieve relevant abstracts from the database into a local database, and from there they operate. They are conducting a couple of mining tasks. They also take into account a set of standard queries, the so-called PICO queries, which is basically a standard set in a standard format relating to specific medical problems. And what you get afterwards is a list, a ranking of which genes and proteins are most relevant to this particular problem. And in the end, again, this cross-validation process. So where comes the Globus and the grid into play? Of course, they use Globus for the job management in order to formulate those mining jobs, to download the abstracts, which means data staging and so on. They use this for the jobs for the semantic analysis and, of course, for storing the data back. All right, so they also conducted a couple of experiments on our IGE testbed. IGE is the initiative for Globus in Europe, which was funded by the European Commission in order to support the Globus activities in Europe. And within the project, we also operated a testbed, and this research was done within this testbed. And in order to get an idea of the performance, they used a standard microarray experiment, which is basically a data set of 98 samples, and they extracted the abnormal genes and proteins, and they ended up with over 40,000 jobs that have been computed. And the average job duration was, if they have the number of abstracts is pretty low, the time for each job is very low, but it can raise up pretty quickly to a lot of more time. All right, so what did they have what experience did they have? First of all, of course, because of the very distributed and parallel processing nature of this task, the grids are the right target environment to do so. And of course, the Globus Toolkit, because it's very powerful in this regard. They had some specific problems. They had to download, for instance, the abstracts for each job from the PubMed database, which imposes a huge workload on the PubMed library service that is actively in use. And so the computing model and the jobs needed to be adjusted in order to have such a flow control in there. All right, so because of the typical grid environment, you end often up with firewall problems, and So, for instance, if you want to access the abstracts from the worker nodes from the local database, you end up with firewall problems, and therefore they use the Globus Online to overcome these problems, which is very cool. All right, so the IGE testbed is, as I already told, ready to use, fully functional GT5 environment, and will also be sustained over the next years. And they are currently evaluating whether this platform can be more integrated with Globus Online and the new service, Globus Genomics, which you will learn more about later on in one of the talks. Yes, and they are also investigating whether it is possible to deploy this service on other platforms, cloud platforms, such as Stratus Lab or the IGE Globus appliances, which is basically a set of pre-configured virtual machines that can run in the cloud, which has been also done as an effort of the IGE project. All right, so the aim of the research was to perform this sort of biomarker discovery and validation process. They have developed this biogene proteomics system, and they are looking towards possibilities to deploy this in the cloud, of course. And there is, I mean, a lot of talks are about sustainability these days, and one possibility here is that a commercial company might be interested in the system. And from what I've heard, they were already successful. There is a commercial company from the omics areas research group in Romania, Austria, and in Germany. So there is a chance that the system can sustain. All right, so let's move on to the second scenario. It was about the dynamics behavior of parallel neural microcircuits simulation environments, which is from the area of computational neuroscience. Computational neuroscience is mainly about understanding how the human brain works. For instance, how the learning works, how the long-term memory works, how information is being encoded and decoded, and so on. But also how the internal structure of our brain works. So there is such dynamic analysis that have to be carried out. And what they simply did is they used different mathematical models that take also the structure and the activation levels and so on of the brain into account. For instance, the Hodgkin-Huxley, the Albert-Kepler, and so on. Applied them on a microcircuit, which is basically a part of the brain, of the human brain, used a specific synapse model, the spike time plasticity thing. And yes, because this environment is highly configurable, you end up with a lot of input parameters, such as the activation currents, the model elements, the values, the geometrical information about the topology, and so on. And then they applied all this on specific simulators, such as the NeuroSim environment, yes, and yeah, did the research. So basically, we end up here with a parameter study with many multi-core codes. We have a lot of independent tasks. We have a high dimensionality of the problem with over 15 different parameters, and we have a large high dimensionality of the results. So this is why the grid is used here as well. And the goal was here to organize the simulations, to manage the results, and to do the post-processing in an appropriate way. And therefore, they developed another framework for the advanced computing investigation of biological neural microcircuits. And here, basically, they put together a set of standard components they developed beforehand. These were Eclipse-plugins mainly, in order to manage the jobs, in order to configure the different scenarios, in order to visualize the scientific results at the end, and so on. And they have also used the grid access layer, which is not only related to the Globus, but also to G-Lite and Saga. Yes, and unless there's a cloud here, the simulation were carried out on the grid. So here is a list of different Eclipse plugins. As you see, we have some for job submissions, file transfer, in order to track the submission history and in order to somehow visualize the results. I would like to mention here that another effort of the IGE project, which is basically the Globus Online client. This is also an Eclipse-based plugin, which covers most of the functionality of the Globus Online, and this is mainly about to show what is possible with the API. So you can use this Eclipse-plugin if you are currently developing a software which is interacting with Globus Online. You can take this as an example to access the grid and Globus Online services, basically. All right, so that's the end of my talk. I hope that it was a bit understandable. Thank you very much for the attention.
All right, any questions? All right, thank you.
Okay, before we break for lunch, I just wanted to acknowledge our gold sponsor, EMC, and give Pat Combs here a few minutes just to walk you through a couple of things. We've just recently started working with EMC on a potential solution that's integrated with their Isilon scale-out storage. So Pat's just going to take a couple of minutes and give us some ideas about that.